Hi, everyone. Thank you for joining. I'm just going to give everyone another minute to join, and then we're going to get started. We're going to have Q&A. So if you want to type your questions on the Q&A box, you can do that, and then I'll answer all the questions at the end of the presentation. So yeah, I'm just gonna go through the content and then at the end answer all the questions you may have. Just gonna give one more minute for everyone to join and then we'll get started. All right. So thanks again, everyone, for joining. My name is Paula Trujillo, and today I'm gonna be talking about neuroimaging studies in multiple system atrophy. I'm a research assistant professor at Vanderbilt University Medical Center. And yes, let's get this started. So this is the agenda for today. First, I'm going to give a quick introduction. Then I'm going to give you a 2-minute lesson on neuroanatomy. And this is going to be useful to understand some of the images that I'm going to be showing during the presentation. Then I'm going to talk about MRI and how it is used in clinical settings to support the MSA diagnosis. Then I'm going to move to more advanced methods that are used in research. The final topic of the talk will be radiotracer imaging methods such as SPECT and SPECT. And then I'm going to finish with some conclusions. So let's start with the introduction. Multiple system atrophy, or MSA, is a rare and rapidly progressive neurodegenerative disorder, which is characterized by symptoms that affect both the autonomic and the central nervous systems. These symptoms reflect the progressive loss of function of nerve cells in the brain. And MSA shares many symptoms with Parkinson's disease, such as slow movement, rigid muscles, and poor balance. The treatment for MSA is aimed at helping individuals cope with some of these symptoms of the disease, but unfortunately, there is no cure for MSA. So what causes MSA? The answer for this is, unfortunately, we still don't know what's the cause for MSA. Some researchers are studying possible inherited components or involvement of environmental factors in the disease process, but today there is no substantial evidence to support these theories. MSA causes deterioration or shrinkage of certain areas of the brain, like the cerebellum, the basal ganglia, and the brainstem, that affect internal body functions and motor control. So under a microscope, the damaged brain tissue of people with MSA show nerve cells that contain an unusual amount of a protein called alpha-synuclein. So in MSA, there is accumulation of this protein. And because both Parkinson's disease and MSA result from an aggregation of these alpha-synuclein protein in cells, they are known as synucleinopathies. There are other mechanisms that have been proposed for possible contributors to MSA, such as mitochondrial dysfunction or inflammation. But again, there are still a lot of things that we completely don't understand about MSA. So as many of you know, the diagnosis of MSA can be very challenging, particularly in the initial stages, as the initial symptoms are often very difficult to distinguish from those of Parkinson's disease. So diagnosing MSA typically involves a combination of medical history, neurological examination, and various tests, such as blood tests and autonomic function tests. There are currently no specific laboratory tests or imaging studies that can accurately diagnose MSA. But while there is no single test to definitely confirm MSA during a person's lifetime, with a complete evaluation by a neurologist that has experience in movement disorders, can help to accurately diagnose based on available clinical criteria and also some test results. Additional tests like brain MRI or a PET scan may be used to rule out or alternative causes, but also it can be used to support the MSA diagnosis. So let's start with our 2-minute lesson on neuroanatomy. So as you know, the brain is a three-dimensional structure. You can imagine your brain as like a piece of bread that you're going to cut into different slices. Each slice gives you a different view of what's inside. So in imaging, we have what we call the neuroanatomical planes. Here on top, we have the sagittal plane, and this gives you a side view of your brain, showing structures from left side and right side. Then here in the middle, we have what we call the coronal plane, and this gives you a view of your brain from the front, showing the structures from the front and the back. And finally, we have the axial or transverse plane. And this gives you a view of your brain from top down, showing structures from above and below. And here we have a representation of the three different planes. So each plane helps doctors and scientists see different angles and perspectives of the brain to understand its structures and how it works. So in MSA, several structures in the brain and nervous systems are affected, which results in the different symptoms. And the most affected structures in the brain include the cerebellum, the brainstem, and the basal ganglia. So let's look at those three in more detail. So the cerebellum, which you can see here in yellow, it's located at the back of the brain, and it is involved in coordinating movement and maintaining balance. Damage to the cerebellum in MSA can lead to problems with balance, coordination, and walking. Then we have the brainstem, which you can see here in blue. The brainstem is a critical part of the brain that connects the brain to a spinal cord, and it controls many essential functions such as breathing, heart rate, swallowing. In MSA, the degeneration of the brainstem can lead to various symptoms, including breathing difficulties, sleep disturbances, and swallowing problems. The brainstem can be divided into the medulla, which is the more inferior part. Then in the middle, we have the pons, and then we have the midbrain. Finally, the other important component of the brain that is affected in MSA is the basal ganglia. The basal ganglia is a group of structures deep in the brain. So here you can see where they are, more or less, what I'm pointing with the cursor. And this is a good group of structures that help control movement. The main components of the basal ganglia include the putamen, which you can see here on this axial slice and here on the coronal slice. We also have the globus pallidus, which is right next to the putamen. And the other important structure is the substantia nigra, which is located in the midbrain. So here, this green line indicates the level where we have the midbrain. And here you can see the axial representation of the substantia nigra. The substantia nigra produces dopamine, which is a neurotransmitter that is essential for controlling movement and is often affected in movement disorders such as Parkinson's disease and MSA. So now that we finish our 2-minute neuroanatomy lesson, we're ready to look at real images. So here we can see an example of an MRI. This is the same image is from just one subject, but you can see the different planes. So on the left, you can see the sagittal plane. In the middle, you can see the coronal plane, and then on the right, you can see the axial plane. And then the red arrow is pointing at different structures. So on the top row, the red arrow is pointing at the cerebellum, on the sagittal, coronal, and axial planes. Then in the middle, we have the brainstem, again here on the sagittal, the coronal, and the axial slice. And finally, we can see the putamen on the sagittal, coronal, and axial slices. So now that you know how to look at MRI images, I'm going to talk about how MRI is used in clinical settings. But before I do that, I want to give you the recipe on how to get an MRI. So the first step to get an MRI is to put a subject in a big magnetic field. The MRI works like a giant magnet. So when you go inside the machine, the magnet aligns the water molecules in your body. Then we apply radio waves that are sent through your body, and this disrupts the alignment. When the radio waves stop, the water molecules realign, and this emits signals. These signals are captured by the MRI machine, and the signals are turned into detailed images of your body's internal structures, which helps doctors to see things inside your body, such as organs, bones, and tissues. We repeat steps 2, 3, 4, and 5 multiple times to get different images, and finally, we allow the subject to leave the scanner. So during the MRI scan, you will hear loud knocking and tapping sounds. These noises are caused by the MRI machine's magnets switching on and off to create the magnetic fields and radio waves that are needed for the scan. So we can produce different images, or what we call different MRI contrast, by adjusting the timing and strength of the radio waves and the magnetic fields during the scan. These adjustments can affect how water molecules in your body behave, which in turn influences how different tissues appear in the final images. So here we can see some examples of images obtained with the MRI scanner. There are 3 main types of MRI contrast. Here on the left, we have an example of what we call a T1 weighted image. These images show the differences in how quickly tissues recover after being disturbed by the MRI magnetic field. Tissues like fat appear bright on these images, and things like fluid, like cerebrospinal fluid, appear dark. So here you can see the cerebrospinal fluid dark in the middle. T1 weighted images are very good for showing anatomy and structures in detail. Here I'm also showing an example of a T2 weighted image. These images highlight how water molecules move within tissues. So in T2 images, fluid appear bright, while dense tissues like bone appear dark. And these images are useful for detecting abnormalities like inflammation or fluid buildup. Finally, we have what we call the proton density images, or PD weighted images. And these images show the distribution of protons in tissues, which helps to highlight the density of tissues and can be helpful for diagnosing certain conditions. So MRI is commonly used to support the MSA diagnosis. And if you go on Google and search for MSA and MRI, this is probably what you're going to find. You're going to see complicated words like hyperintensities, which actually means that there are areas that look bright in the images. You're going to see the word hypointensities, which means that there are areas that look dark. You may also see these words on the MRI report from your neuroradiologist. Another word that you may see on the reports is the word atrophy, which means that there are areas of the brain that are shrinking. So if you continue to search online, you may find things like you see on this slide. There are some common MRI features that may be observed in cases of MSA. And these include atrophy, or as I said, shrinkage of areas in the brain like the cerebellum, the brainstem, and the basal ganglia structures, particularly the putamen. You're gonna see the word hot cross bone sign, which is a distinctive imaging feature seen on axial MRI slices of the pons. This sign is not specific to MSA, but is commonly associated with MSA, particularly the MSA-C subtype. There are signal changes in the putamen, which are often described as hypointensity, or again, darkening of this region. And there are other modalities like diffusion-weighted imaging that can also be used to assess the movement of water molecules in tissues, which can provide information about certain changes happening in MSA. So here on this figure, you can see, remembering from your neuroanatomy class, the sagittal view of the brain, showing atrophy of the cerebellum and the brainstem. And here you can see the hot cross bone sign on the axial slice of the pons. So I'm gonna show you a couple of typical cases. So on the first case, we have a 50-year-old male patient with progressive ataxia and gait dysfunction, which means that this person had trouble walking steady and smoothly. This patient also had dysarthria, which means that the patient was having difficulties speaking clearly. Dysphagia, which means that he had difficulties swallowing. And the MRI scan show the hot cross bone sign and also dark areas in the brain in the basal ganglia, especially the globus pallidus. So here on the axial slice of the pons, we can see the hot cross bone sign, and on the axial slice of the basal ganglia, we see this dark area on the for the globus pallidus. Here's another case. This is a 65-year-old female patient with predominant Parkinsonian symptoms. And the clinical MRI show the T2 hypointensity within the putamen, which was more evident than the globus pallidus. And it also showed what's known as the putamenal rim sign, which just means that there is a dark outline around the putamen. So again, here is a coronal view of the putamen, and this dark area here is the putamen, and here are axial slices at the level of the putamen showing that hypointensity in that region. So MRI plays a critical role in supporting the diagnosis of MSA. Structural images can provide detailed information about the brain and can allow doctors to examine structures for any abnormalities. As I said, in MSA, there are specific patterns of atrophy and changes in specific brain regions that can be observed, especially in the putamen, cerebellum, and brainstem. MSA affects not only these areas, but it also affects white matter, which are the nerve fibers that transmit signals throughout the brain. And these changes in white matter may also be associated with MSA. MRI is also very valuable in ruling out other conditions that may mimic MSA, such as Parkinson's and progressive supranuclear palsy, or PSP. And by carefully analyzing MRI findings alongside clinical symptoms and other clinical tests, doctors can make a more accurate diagnosis of MSA. So to summarize, these are the two most classical signs observed in MRI. On the left, we can see the hot cross bone sign at the level of the pons. This is typically more associated with MSA-C. And on the right, we see what's known as the putamenal rim sign, or darkening of the area of the putamen. And this is typically more associated with the MSAP subtype. And while these signs can help diagnose MSA, they are not always conclusive. And they may be seen in other conditions too. Additionally, a combination of these signs can be seen in the majority of MSA patients at different stages of disease. So they may not be evident at initial stages, but eventually at different stages, these may become more apparent and independent of the clinical phenotype. But here is the first conclusion of this talk is that one size doesn't fit all. MSA is a complex and highly variable condition, meaning that it can look very different from person to person, and this variability extends to how the disease appears on the MRI scans. For example, one person with MSA may have more trouble with movement, while other might have more problems with balance or speech. And the severity of symptoms can also vary widely from person to person. So while MSA is the same condition, it can look very different depending on the individual. This means that the abnormalities or unusual findings on the MRI can vary from patient to patient. Some may show certain patterns, while others may have different ones. So again, it is important, MRI is important, but it must be considered alongside symptoms, disease progression, and treatment response for a complete understanding of MSA. So to finish this section, I just want to show you some examples of a T1 weighted MRI for 3 different people. On the top, we have a 55-year-old female with no neurological disorders. On the second row, I'm showing the T1 weighted images for a patient diagnosed with MSA-P. And this image reveals atrophy of the putamen, which you can see here on the axial slice. So you see that the putamen is smaller compared to the healthy person. And lastly, on the bottom, you see the MRI scan of a 59-year-old man, also diagnosed with MSA, but he had the MSA-C subtype. And in this image, atrophy is observed in the cerebellum, while the putamen is mostly preserved. So while MRI is a valuable tool, it has several limitations when it comes to diagnosing MSA. MRI findings in MSA can overlap with other disorders like Parkinson's disease or PSP. So it is challenging to differentiate MSA. MRI might not be sensitive enough to detect subtle changes in certain brain areas affected by the disease, especially in early stages. So the diagnostic accuracy of MRI might vary a lot based on the stage of MSA. As the disease progresses and more substantial changes occur in the brain, MRI might become more informative, but this may be too late for intervention. So in the early stages of MSA, MRI might not show significant abnormalities, and this can lead to misdiagnosis or delayed diagnosis. While MRI can provide detailed structural information about the brain, quantifying the extent of degeneration or changes specific to MSA can be difficult, and this can hinder tracking disease progression accurately. MRI primarily provides structural information about the brain, but it might not capture the functional aspects of the disease, which are equally important for diagnosis and also understanding the disease impact. And finally, different imaging centers might use different MRI protocols, and this can lead to variations in image quality and interpretation. So alongside conventional MRI techniques, researchers are exploring novel methods to investigate MSA more comprehensively. And these advanced imaging techniques aim to provide deeper insights into the underlying pathology and progression of the disease. These emerging imaging techniques complement conventional MRI, but they offer new opportunities for understanding the pathology of MSA and provide the opportunity to identify disease biomarkers and develop target treatments. And while these are still under research, these advanced imaging methods hold promise for improving diagnosis, management, and future therapeutic interventions. So I'm going to describe a few of these advanced methods. These are known as quantitative MRI because they go beyond traditional qualitative imaging, and they provide a quantitative measurement of different tissue properties, and they can offer valuable insights into structural but also functional changes within the brain. Quantitative MRI can be ideally comparable across imaging sites, different time points, and different acquisition methods. So they can be used to track disease progression and also to evaluate the effectiveness of therapeutic interventions. And in fact, the failure of previous therapeutic trials in MSA has been in part attributed to the enrollment of participants with advanced disease, which are unlikely to benefit, experience a benefit due to the severity of the pathological process. So it is very important that these quantitative imaging markers can help with the patient stratification and can be used as biomarkers in clinical trials to understand how these treatments are affecting the brain. So the first method that I'm going to talk about is MRI volumetry. This is a method that is used to measure the volume of specific brain structures or regions from the MRI scans. In this approach, expert neuroradiologists carefully delineate the boundaries of structures in the brain, including the putamen, the cerebellum, and the brainstem. And while this is considered the gold standard for MRI volumetry, manual segmentation is very labor intensive, it is time consuming, and is also prone to inter-rater variability. So recent technologies and discoveries have allowed the development of automatic methods. These are also known as computer-assisted segmentation approaches, and these use algorithms and software to automatically segment brain structures from MRI images. These algorithms may rely on intensities, edge detection, machine learning, or other approaches to identify and delineate structures. These are faster and more consistent than manual segmentation, but they may require some manual correction and validation to ensure the accuracy. Finally, there are other studies that have used what's known as voxel-based morphometry, which is a computational technique that is used to analyze different differences in brain anatomy between groups or of individuals. So using this method, for example, we can compare patients with MSA versus patients with Parkinson's disease or healthy individuals. And this involves preprocessing the MRI scans to align them into a common space, followed by a statistical analysis to identify regions with significant volume differences. Recent studies suggest that MRI volumetry can be used to track changes in brain volumes over time, even in early stages of MSA. And these findings indicate that MRI volumetry could be a useful tool for monitoring disease progression and also evaluating treatment effectiveness in MSA patients. So here we see a figure from a recent publication showing how the volume changes over time are different between MSA-C and P for different brain regions compared to healthy control. So automatic segmentation methods in MRI volumetry are often trained using images from normal controls to learn the typical appearance of brain structures. But these methods may encounter challenges when they are applied to pathological conditions such as MSA, where there are diverse anatomical changes that can alter how the images look. So to address this limitation and improve precision, researchers need access to more data, including MRI from patients with MSA and other neurological conditions. So we need more data because to improve the accuracy, we need to train these algorithms with images that look like the images that we obtain from patients. So this will allow us to refine the algorithms and develop specialized models that are specifically tailored to MSA characteristics and can further enhance segmentation accuracy and reliability. And now I'm gonna move to diffusion-weighted imaging. This is a technique that examines how water molecules move within brain tissues. In MSA, it can reveal alterations in the microstructure of white matter pathways, which are the brain's wiring responsible for transmitting signals. Changes in these diffusion patterns, particularly lower values of what's known as fractional anisotropy, or FA, or higher values of a metric that we obtain called diffusion mean diffusivity, or MD, could indicate disruptions of these pathways, suggesting damage or degeneration. And by providing insights into the integrity of the white matter tracks, diffusion imaging could assist in diagnosing MSA and also monitor disease progression over time. Another important quantitative imaging methods are those that are sensitive to iron deposition. Understanding how iron works in the brain is complicated and still not fully known. But we do know that iron is very important for keeping brain working normally. When iron levels are elevated, it can harm the brain in different ways. Too much iron is linked to many diseases, including MSA. As we get older, our brains tend to collect more iron, and this is more particular in certain brain regions like the substantia nigra, the putamen, the globus pallidus. So why do we care about measuring iron in MSA? Even though it is not exactly sure how MSA starts, there is evidence to suggest that iron plays a big part of it. Iron seems to promote alpha-synuclein accumulation and can cause damage in key parts of the brain. In MSA, along this accumulation of alpha-synuclein, there are other things happening simultaneously, such as iron accumulation, loss of brain cells, inflammation. And studies have found that higher iron levels in specific brain areas of people with MSA can be observed in MRI scans. How does it look? So on MRI scans of people with MSA, we can see this iron accumulation as darker areas on T2 images. There are also more specialized sequences known as susceptibility weighted images, or SWI. You also see the areas with high iron content as darker areas. SWI is really good at finding changes in the brain caused by iron accumulation, especially in the putamen, and it's better than just T2 imaging for this, but it can't give us exact measurements. So that's where quantitative methods come in. There's a new method called quantitative susceptibility mapping, and another method called R2-star mapping that can tell us how much iron is in the brain. So studies that have used these methods have seen higher iron content in patients with MSA. So here are other examples of QSM images from a control patient with Parkinson's disease and patients with different stages of MSA. The higher the susceptibility values, the more iron it is. So the areas that are red in these images are areas that have more iron content. So we have used these methods, and we have looked at how much iron was in certain parts of the brain and how bad the symptoms were in patients with MSA. We have used QSM to measure iron levels in areas like the globus pallidus, the putamen, the substantia nigra, and the dentate nucleus. And we compare the iron levels between different groups of people and check if that was associated of how severe the disease was. We found that compared to people with Parkinson's disease and also those with any brain problems, people with MSA had more iron in the putamen, globus pallidus, and substantia nigra. And they also had higher iron levels in a structure in the cerebellum called the dentate nucleus. So people with MSA have more iron in these structures, and this was also associated with worse symptoms. So these findings suggest that having too much iron in certain parts of the brain might make MSA symptoms worse. Here's another example, two different patients, one showing iron accumulation in the cerebellum. So here's the dentate nucleus of the cerebellum. Then we have the substantia nigra in the midbrain, and then this patient didn't have iron in the putamen. Whereas this other patient, the iron content in the dentate was lower, but this patient did have high iron content in the putamen. So these images can also be informative about the different subtypes of MSA. Okay, moving to another method, we have what we call functional imaging, also known as resting state functional MRI. And this measures brain activity at rest. So while the patient is resting at the scanner, we measure the signals and we try to understand how different brain areas are working together. So by analyzing these connections, resting state functional MRI can provide insights into disease progression, severity, and treatment effects. There are different networks in the brain. Those are known as the default mode network, but there are other networks like the sensory motor, visual, the language network, the attentional network. So all these different networks can be affected in MSA, and here is a figure showing areas in MSA that are different compared to normal controls. Then we have another technique called magnetic resonance spectroscopy. With this method, we do not obtain an image. We do not get like a picture of the brain, but we get what we call a spectrum. So it looks like this, and each of these peaks is associated with a different metabolite. So this allows us to estimate concentrations of different metabolites non-invasively. These different metabolites indicate different neuronal function or tissue changes. For example, the metabolite called myoinositol can be informative for gliosis, whereas another known as N-acetyl aspartate or NAA can inform us about neuronal integrity. So during the MRS scan, signals from metabolites are acquired and identified based on their chemical properties. So the different metabolites resonate at different frequencies, and we use this information to estimate the metabolite concentration in the brain. Finally, we have other methods that allow us to assess cerebral blood flow. So we have a method that's known as arterial spin labeling or ASL. And this works like a special camera that takes pictures of your brain. But instead of using contrast agents, it labels the blood in your arteries with a magnetic signal. This blood travels to the brain, and then we take pictures with and without the labeling and understand how your blood is flowing through the brain. And here on the right, I'm showing an example from one of our studies where we use this method to measure changes in blood flow in patients with Parkinson's disease and understand what was the effect of medication. So we scan patients off their medication and on their medication and see how blood flow was different, to see the effect of this medication. As far as I know, only a few studies have used this technique in MSA, but this technique is very promising because it provides the opportunity to non-invasively measure brain function. And I know that ongoing studies are including these methods in their imaging protocols. So I'm just going to spend the last couple of minutes talking about PET. So I gave you the recipe for MRI. I'm gonna give you the recipe to obtain the PET images. So PET stands for positron emission tomography. And this works very different to the MRI. These images are more invasive because they involve the use of a radio tracer. So the first step to obtain the PET images is the tracer injection. So a radio tracer is injected into the body, which is a biologically active molecule labeled with a positron-emitting radionuclide. This tracer circulates through the bloodstream and accumulates in specific tissues or organs based on the chemical properties. This tracer emits positrons that annihilate with electrons, producing gamma rays. And the PET scanner detects these gamma rays, and this information is used to reconstruct information about function, and particularly metabolic chemical activity in the body. The SPECT, which you may also have heard, is similar to PET, but the tracer that it's using SPECT emits gamma rays directly upon decay without the involvement of positrons. But both techniques provide functional rather than structural information, making them particularly valuable for studying physiological and biochemical processes. So the most common radio tracer imaging that different studies have used to investigate MSA are dopamine transporter. Using these tracers, dopamine transporters are target, and it can provide information about dopaminergic dysfunction, which is a characteristic of MSA, particularly the MSA-P subtype. There are also PET scans that are informative for the glucose metabolism. So scans known as FDG-PET detect patterns of reduced glucose uptake in affected brain regions. There are also other PET methods that are more sensitive to microglia activation, so neuroinflammation. And here on the right, you see an example of these images. So each of these techniques provides a unique insight into MSA pathology, which can support the diagnosis and provide also information for prognosis and treatment development. So to conclude, significant progress has been made in neuroimaging techniques for studying neurodegenerative diseases. In MSA, most of previous studies mainly focus on volumetric and structural changes in the brain. But novel methods are also focusing on connectivity and brain function, and these can improve diagnostic accuracy and identify novel markers for disease progression in MSA. These novel imaging biomarkers can improve patient selection in clinical trials, and has the potential for assessing treatment-induced changes. But no single technique can provide all necessary measurements, and combining multiple technologies in a multimodal approach is the most promising path for future progress in clinical diagnosis and MSA research. So we need additional data from MSA patients. Gathering more data will enhance our comprehension of this complex disease. I see MSA as a highly complex puzzle, and as we accumulate more pieces, a more clear understanding of the complete picture. So if you're interested in participating in research studies, please contact Jessie Iregui. And I thank you for your attention. And here is the picture of my lab at Vanderbilt. So now I'm open to questions. I will stop sharing and I'll go to the Q&A. The first question says, at what stage in the progression of symptoms in MSA will you begin to see changes on MRI? And this is an excellent question. There are several, so in structural images that are used in clinical practice, it's more challenging because it's more qualitative, and if the changes are not very evident, the neuroradiologist might not be able to detect subtle changes. But there are still things that can inform about the presence of MSA. So if a patient presents with symptoms that are, more cerebellar, and then the MRI shows cerebellar atrophy, it can be used to support the diagnosis. Novel methods such as the QSM that are sensitive to iron deposition are also showing that there are changes that are evident in early stages of disease. So we have conducted a study for early MSA patients, and we have seen changes in iron content in different structures of the brain, even at early stages. So the novel methods that are being developed, particularly those that provide more quantitative information, will be more sensitive to detect changes in early stages. So the next question is, can you address MRI findings when clinical symptoms are a mix of MSAP and MSAC? Yes, this is a great question, and what clinicians are seeing as a mix of MSAP and MSAC may also be a mix on MRI. Like sometimes patients have changes in different places, like the cerebellum, the brainstem, and the basal ganglia. For the MSAC, most of the changes occur in the cerebellum and the brainstem. And for MSAP, most of the changes occur in the basal ganglia, particularly the putamen. But there is everything in between. Like some patients may only have changes in the cerebellum, while others may also have changes in the putamen. But there are patients that may have a mix of everything. So MRI can also be very informative in these complicated scenarios where the clinical manifestation does not provide a clear phenotype, whether it's MSAP or C. And we in our lab believe that this is not black and white. There is not just like two subtypes. It's a spectrum. Like we see a mix of findings also on MRI. So the next question is, does cryo-EM will improve the diagnosis and research in the future? Unfortunately, not familiar with this technique, and I'll be very curious to read more about it. So yes, I can provide an answer for that question. The next question is, I understand that iron deposits are found in the brain, but it will be a hint if someone has iron infusion to bring their level up to normal levels, but this decreases quickly. Iron level decreases, but ferritin staying stable. So if I understand this question correctly, it might be related to taking iron supplements or iron infusions, but it doesn't necessarily be related to what's going on in the brain. The accumulation of iron in the brain occurs with aging, but also in certain conditions like MSA, there are things that could contribute to iron accumulation. some of them may involve disruptions in the blood-brain barrier permeability, iron dyshomeostasis. So there are things that can contribute to iron deposits in the brain, but I don't think having like iron infusions could be related to what we're seeing in this condition. Okay. The next question is, can you explain the F-DOPA scan? I was given the radio tracer and Cinemat for that scan, but I didn't understand the use of Cinemat for this test. So yes, so the F-DOPA scan provides information about dopaminergic function in the brain. I'm not sure if you were taking Cinemat as part of your like regular treatment, but before the and I'm also not sure if the purpose of giving you Cinemat for the scan was the levodopa that comes in the Cinemat, or was more the carbidopa that is also present in this medication. I will guess probably the purpose of giving you Cinemat was to use the carbidopa to avoid the to avoid the metabolism of the F-DOPA tracer within the body, and it allows to go for this radio tracer to go into the brain. So yes, I think the goal of giving you Cinemat was to avoid the radio tracer going into the body and directing this radio tracer to the brain. What is a DAT scan? Yes, that's another question that is also related to dopaminergic imaging. So DAT scan is a scan that involves a radio tracer that binds to dopamine transporters. It is used with a combination of this radio tracer and a SPECT acquisition. So this is used with a SPECT scan. So this tracer will emit gamma rays that are detected by the SPECT scanner, and this allows some information about dopaminergic function in the brain. Typically in conditions that affect dopaminergic function, like Parkinson's disease and also MSA, particularly the MSA-P subtype, you can see decreases of the dopamine transporter binding. This is what we call when the radio tracer goes to the areas of the brain that we want it to go. So we see decreases of the signal in specific brain regions, such as the putamen. So in a DAT scan that shows low signal in the putamen may suggest lower or impaired dopaminergic function. But unfortunately, it is not easy to distinguish MSA and Parkinson's from these scans. So the next question is, will the slides be able to be viewed individually, as they are very helpful in understanding the information? I will probably pass that question to Mission MSA, which is gonna make this content available online. So I'm not sure if the slides will be available or just this webinar as a whole. So yeah, they might be able to answer that question. Next question. My husband takes an iron supplement every day. Should we discontinue it? I will say no. He's probably taking iron supplement because he may have anemia or other conditions. And if the doctor recommended taking iron supplement, I think it is very important for him to continue taking this supplement. Iron accumulation in the brain, I think, is happening because of complete different mechanisms. So yeah, I wouldn't recommend to discontinue it. So the next question is, what do you think from a research perspective is the most important thing clinicians can do to decrease the misdiagnosis of MSA and Parkinson's disease? So the focus of today's webinar was on imaging biomarkers. There are several of these that I think are gonna be incorporated in clinical practice, hopefully in the near future, that can provide more quantitative information about these conditions. But in addition to the imaging biomarkers, there are also fluid biomarkers. So there are things that we can measure from the blood, as well as things that we can measure from cerebrospinal fluid, that can provide more information. And I think the combination of imaging biomarkers as well as fluid biomarkers together with the complete clinical evaluation is what's going to be more useful for clinicians to decrease the misdiagnosis. Currently, the fluid biomarker seems very promising in distinguishing MSA from Parkinson's. So I think that will also be translated into clinical practice, hopefully not very far from now. So the next question: for a DAT scan, a patient was instructed to hold his normal nightly medication for 48 hours. It was held for 36 hours. Will this invalidate the exam? I don't think so. This ideally you will like to follow the instructions from your clinicians, but the lifetime of these medications are typically less. These instructions tend to be conservative, so I will probably think that this shouldn't invalidate the exam. Someone just follow up on the previous answer, and it's asking, what are the biofluid markers you just mentioned in helping clarifying the diagnosis? So there are different things that you can measure from blood and also cerebrospinal fluid. One of those is known as NFL, and that can provide it's like, we think higher levels of NFL are associated with axonal damage, and several studies, including a research study from Mayo Clinic, show that the levels of NFL are very different between patients with Parkinson's and patients with MSA. So I think that that's something that could potentially be translated into clinic. There are other things that we can detect from cerebrospinal fluid, such as the one is known as alpha-synuclein aggregation assay, and this one can provide information about alpha-synuclein aggregation, and in PD and MSA there are different profiles, so that could also help for the differential diagnosis. I think I'm gonna answer the last question. So since Cinemed isn't very effective with MSA, why take it if there is no reaction to taking it? I'm not sure if you're talking about like taking the medication on a daily basis to treat symptoms, or if it was prescribed prior to a scan. So it will depend on that, but I will follow your clinician's recommendations. So thanks again, everyone, for joining. This webinar will be available online, and Mission MSA will provide more information about it to all the attendees. Thank you.